Welcome to Biology with Mrs. Ignacio. We are going to get into lesson 3A and then lesson 3B. So this lesson starts off with the discussion of cytology. Remember, cytology is the study of cells. It's also known as cell biology. The cell is the smallest unit of life itself. So it is actually living. Again, cytology is the study of cells or the study of cell biology. So let's talk about the discovery of cells. Robert Hooke was the very first person to see a cell using a homemade microscope. Hooke was a contemporary of scientist Isaac Newton. Contemporary means that they lived at the same time, during the same time period. So Robert Hooke was the first person to see a cell using a homemade microscope, which is pretty amazing. Now, Robert Hooke was a contemporary of the scientist Isaac Newton, meaning that he lived during the same time period as Isaac Newton. Matthias Schleiden was a German botanist who learned that all plants are composed of cells. So a botanist is a scientist that studies plants, trees, all of those kinds of things, flowers. So Matthias Schleiden was from Germany and he was a botanist and he learned that all plants are composed of cells. And he probably was able to see the plant cell that had a cell wall, then the cell membrane, and all the organelles that lie within the plant cell. So that's pretty neat. We have Theodore Schwann, who was also a scientist, and he was determined that all animals were composed of cells. And he's absolutely correct. So Theodore Schwann was a scientist who was able to determine that all animals are composed or made up of cells. Rudolf Virchow was a scientist in 1955, proposed that all cells come from pre-existing cells, meaning that cells that have been alive before. So that is Rudolf Virchow. He was a scientist in 1855. He proposed that all cells come from pre-existing cells. And that's going to be important because that leads us to our cell theory. The cell theory is something that you must know. And if you're taking notes, highlighting, this would be one of the things that I would highlight in my notes. So the cell theory has three basic principles. The first is that all living things are composed of one or more cells. The second principle in cell theory states that a cell is the basic unit of organization in all organisms. When we think about the basic unit of organization or the level of organization, we always start from the smallest to the largest. So if you have a cell, the cells combine to form tissues. Tissues combine to form organs. Organs combine to form organ systems, and organ systems combine to form an organism. So cells make tissues, tissues make organs, organs make organ systems, and organ systems make an organism or a living being. Okay. So the third theory of cell theory, or the third principle rather of cell theory, is that all cells come from existing cells. So a cell had to be alive in order to produce another cell. So these are the three basic principles of cell theory. All living things are composed of one or more cells. A cell is the basic unit of organization in all organisms, and all cells come from pre-existing cells. When we think about modern cell theory, we have some more observations about cells. And this is also another important kind of point to note, to highlight, star in your notes. Cells use and transfer energy, so they pass energy along. So cells use and transfer energy, and hereditary information is passed from cell to cell. And we know that hereditary information is in the form of DNA, deoxyribonucleic acid, that is found in the nucleus of the cell. All cells have the same basic chemical composition. And we're going to actually zoom in and look at this composition in just a few moments. So with modern cell theory, we know that cells use and transfer energy. Hereditary information is passed on from cell to cell, and all cells have the same basic chemical composition. So when we talk about the levels of organizations or the organization of cells, we are looking at multicellular organisms. So from a cell, we know that cells form tissues, and a tissue is a group of specific cells that are connected or joined together to carry out a specific function or a specific task or a specific job. Now we look at these tissues that form organs. Organs are several types of tissues that work together to perform a function. So we have tissues, then we have organs, and then these organs work together to form an organ system. A group of organs cooperating together to perform a function makes up a system. So if you have your mouth, your esophagus, your stomach, your small intestine, your large intestine, these all work together to make up the digestive system. And the digestive system does perform a function. The function is to break down food into fuel that the body can use. So now let's talk about the two different types of cells. We have prokaryotic cells and eukaryotic cells. We're going to talk about prokaryotic cells first. So prokaryotic cells are single-celled organisms, meaning that they're just one. Examples of prokaryotic cells can be bacteria and green algae. These prokaryotic cells have a cellular membrane around the cell, but they do not have a nucleus or any membrane-bound structures inside the cell. So they are very simple, very basic kind of organisms. Again, this is the first type of cell. This is a prokaryotic cell. They are single-celled or one-celled organisms. Examples of these prokaryotic cells could be bacteria and green algae. Prokaryotic cells have a cellular membrane around the cell, but no nucleus or membrane-bound structures inside the cell. The second type of cell is a eukaryotic cell. An eukaryotic cell is the type of cell that you'll find in multicellular organisms. Guess what? Humans are multicellular organisms. Multi meaning many, cellular meaning cells. So humans are a great example of multicellular organism. And yes, we are made of eukaryotic cells. So the examples listed are plants and animals. So the two types of cells, we have prokaryotic cells and then eukaryotic cells. Let's take a closer look at eukaryotic cells. We're going to look at the structure of eukaryotic cells. So let's talk about from the outside in. So eukaryotic cells have a plasma membrane, also called a cell membrane. So the outer boundary of the plasma membrane is actually the outer boundary that separates the cell from its environment. So consider the plasma membrane as the gatekeeper. It lets some things in, lets some things out. Again, the plasma membrane, also known as the cell membrane, is the outer boundary that separates the cell from its environment. Now we're inside the cell, and inside the cell is a substance that is called cytoplasm. So the cytoplasm consists of the fluid that's inside of the cells, and it also contains molecules and organelles. And we're going to talk about these different kinds of organelles and what their jobs are. But remember, the cytoplasm is the fluid inside the cells, and it contains molecules and organelles. When we think about the cytoplasm, I want you to think of your favorite soup. And maybe your vegetables, your meatballs, maybe the carrots, whatever you like to put in your soup are going to be the different organelles. The broth or the liquid is going to be what's called cytosol, okay, cytosol. So the broth of your soup is cytosol, the meatballs, the pasta, the vegetables, the potatoes, whatever you put in your favorite soup, that's going to be the organelles. Okay, so organelles are known as little organs. Organelles are little organs that are inside the cell. So the nucleus is the control center of the cell. It contains genetic information, the DNA, deoxyribonucleic acid. So that is really important to understand. The nucleus is the control center of the cell. It contains DNA, the genetic material of the cell. That concludes lesson 3A. Let's jump into lesson 3B, where we talk about the eukaryotic cells and the main parts of the eukaryotic cell. This is also a research question. Many of you missed the research question. So this is the answer to that research question that you had for lesson 3. The 3 main parts of the eukaryotic cell will be the plasma membrane, the cytoplasm with organelles, and the nucleus. Let's take a deeper dive into these 3 main parts, the plasma membrane, cytoplasm with organelles, and the nucleus. And we're still discussing eukaryotic cells. So let's look at the plasma membrane. The plasma membrane, also known as the cell membrane, has a really important job. And that really important job is maintaining homeostasis. Remember, homeostasis is an internal steady state, okay? Keeping everything at normal levels. So how does this plasma membrane or cell membrane keep homeostasis? Well, the membrane does this through what's called selective permeability. It is very selective on what it lets in and very selective on what it lets out. So this means that it allows certain things in and it keeps other things out. Okay? So what is this plasma membrane composed of? Well, that's a great question. The plasma membrane is made of what's called a phospholipid bilayer. Okay, so this phospholipid, let's break up the word. Phospho means that it has some phosphorus or phosphate groups. Lipids are fats. Bilayer, bi means 2, so there are 2 layers. So this phospholipid bilayer has is what makes up the cell membrane. And let's talk about phospholipids a little bit. Phospholipids have a hydrophilic, philic meaning love, and hydro meaning water. So water-loving head. So this phospholipid bilayer has hydrophilic, which is a water-loving head, and that's the circle that you see in a lot of the models. And then it also has 2 hydrophobic. Remember, hydro is water, phobic is fearing. So 2 hydrophobic or water-avoiding tails, and those are the long squiggly lines that you see in the models. So again, phospholipids, we have hydrophilic, water-loving head, and then 2 hydrophobic, water-avoiding tails. And this structure forms the bilayer. And bi is 2, like a chocolate layer cake, right? It has 2 layers. So you can say that your chocolate cake, your German chocolate cake, maybe your angel food cake is a bilayer. So this bilayer structure is also called a fluid mosaic model. When we think about the fluid mosaic model, it helps us understand what the cell membranes are and what they do. So the cellular membrane is not a solid barrier, but it moves lipids like a fluid, right, with cholesterols, protein, and carbs, which is short for carbohydrates, that are embedded in the membrane. So this fluid mosaic model was proposed by S.J. Singer and Garth Nicholson. This model, this fluid mosaic model, explains the structure of the plasma membrane of animals. And it says that the membrane is a mosaic. It's like a work of art that is composed of phospholipids, proteins, cholesterol, carbohydrates. And these components give a fluid character to the membranes, meaning that things are able to move and shift throughout the membrane. Okay. So that is your fluid mosaic model. Okay. So let's talk about the components of the cell membrane. We have cholesterol. Cholesterol is important because it stabilizes the membrane. It helps to prevent phospholipids from sticking together. And also in the membrane, there are transport proteins. They form channels for specific molecules to enter and leave. It's kind of like a nice doorway for these molecules to enter and leave. Also in the cell membrane, you will find proteins and carbohydrates. And these proteins and carbohydrates are important because they identify the cholesterol and transport proteins as they come in and out of the cell. So again, the components of the cell membrane, we're looking at cholesterol, transport proteins, other proteins, and carbohydrates. So molecules are able to move through the cell through active and passive transport. So passive transport does not require energy, but active transport does require energy. Okay, so passive transport does not require energy, while active transport does require energy. So when we think about passive transport, we are moving ions and molecules across the cell membrane, again, without requiring energy. We have diffusion, which falls under passive transport, and molecules will move from an area of higher concentration to lower concentration. And this is Brownian motion. So this is what we see if we were to look closely at these molecules in a microscope. So it's kind of a random movement of these microscopic microscopic particles in a fluid. That's Brownian motion, the random movement of microscopic particles in a fluid. Okay. Also, what falls under diffusion is the fact that diffusion is affected by temperature. It's also affected by the concentration and the size of the molecules. So you would imagine most times if we heat something up, the molecules will move faster. If the temperature is cooled down, those molecules may move a lot slower, okay? So passive transport does not require energy. An example of passive transport is diffusion, where molecules move from an area of high concentration to lower concentration. So let's take a look now at osmosis. This is still falling under passive uh, transport, but osmosis is similar to diffusion, but with the solvent diluting the solution. Most times, the solvent that we're talking about is going to be water. Okay, so osmosis is similar to diffusion, but we're looking at the solvent diluting the solution. And again, when we're talking about passive transport, molecules are moving from a higher concentration to a lower concentration to try to get to an equilibrium. Now, let's talk a little bit about active transport. Active transport is the movement of molecules across a membrane from an area of lower concentration to a region of higher concentration against the concentration gradient. So this is going to require energy to actually push those molecules into an area that has higher concentration. So when we think about active transport, again, I want you to understand that we're using energy to move molecules from a higher concentration, from a lower concentration to a higher concentration. So it's the exact opposite of passive transport. And in order to get these molecules to move from lower to higher, it's going to require some energy. So, for example, if you were living on a big farm out in the middle of the country and your family said, okay, we're going to move you to New York where everybody is kind of crammed in a small space, you're going to really need a lot of energy in order to make that move. That is active transport because in the farm area, the land is spread out. There's not that many neighbors for miles to see. But if you were to move to New York City, people are stacked kind of in buildings. They're in close proximity to each other, and that's a higher concentration of people. So that's kind of an example that you can use to understand active transport when we have molecules moving from a lower to a higher concentration. So we use transport proteins to actually help these molecules to enter and to leave, to actively transport them. So there are specific proteins that form these channels to help the molecules enter and leave. Still talking about active transport, we have to look at a few things: endocytosis and exocytosis. Endo, I want you to think inner, and endocytosis, right? Cytosis is movement of the cytoplasm or movement of the cell. Endocytosis is movement into the cell. So things are moving into the cell via endocytosis. This could happen in two ways. We have phagocytosis and pinocytosis. So remember, endocytosis, things are moving into the cell. If we have phagocytosis, this is a cell taking in solid material. I think of it kind of like Mrs. Pac-Man or Pac-Man, chomping, 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 right? So this is phagocytosis, a cell taking in solid material. Now, pinocytosis is a cell taking in liquid material. So if Pac-Man or Mrs. Pac-Man is chomping on a nice meal, they need to wash it down with some water, and that's where pinocytosis takes place. So pinocytosis is the cell taking in liquid. Phagocytosis, solid material coming in. Pinocytosis is liquid material coming in. So now that we've taken in all of this kind of material, both solid and liquid, we have to look at exocytosis. Exo, I want you to think outer, just like some insects have an exoskeleton, meaning that their skeleton is on the outside. When we think of exocytosis, this is moving of some molecules out of the cell. And most times this will be your waste materials, right? So endocytosis, endo in, exocytosis, outer, moving out of the cell. All right, let's take a look now at cytoplasm and organelles. Cytoplasm is the fluid that fills the space of the cell, and that is called cytosol. So the liquid portion of the cytoplasm is called cytosol. So if I'm using my soup analogy, the cytosol would be equal to my chicken broth or the broth of my soup, okay? So the cytoplasm, think of it as your soup. The liquid or the broth portion is going to be your cytosol. So when we think about the cytoplasm has what's called a cytoskeleton. And the cytoskeleton is a network of fibers that help to give the cell its shape, just like our skeleton helps to give our body's shape. The cytoskeleton gives the cell its shape, and it is a network of fibers. Organelles, they are little organs inside the cells, and they interact with each other. These organelles function or work to keep the cell running smoothly. So organelles are little organs inside the cell, and they do interact with each other, and they function, or their job is to keep the cell running smoothly. So let's take a closer look at the different kinds of organelles. So let's talk about ribosomes first. Ribosomes are the smallest and the most numerous organelles in the cell. That means there's so many of them. And so these ribosomes, again, are the smallest and most numerous organelles in the cell. They are made inside the nucleus of the cell. Remember, the nucleus is the control center or the brain of the cell. Ribosomes are made of proteins and strands of RNA. RNA stands for ribonucleic acid. The job or the function of ribosomes is to line up these amino acids in order, and they create different proteins. So when you think about ribosomes, you should think proteins. Now, ribosomes can float freely in the cytoplasm, or they can be attached to the endoplasmic reticulum. Again, ribosomes can float freely in the cytoplasm, or they can be attached to the endoplasmic reticulum. So let's talk about this endoplasmic reticulum. If we have ribosomes attached to the endoplasmic reticulum, it is known as rough endoplasmic reticulum. So rough endoplasmic reticulum has ribosomes attached to it, making it look bumpy. I challenge you to, with the permission of your grown-up, to look up what each of these specific organelles looks like. Take a search, a Google search, to see what they look like. When you look at the rough endoplasmic reticulum, you will see that it has little ribosomes and it looks bumpy. The job or the function of the rough endoplasmic reticulum is producing proteins. So the rough endoplasmic reticulum has ribosomes, and its job or function is producing proteins. What about the smooth endoplasmic reticulum? Well, guess what? Smooth endoplasmic reticulum does not have ribosomes attached. It appears to have a smooth surface. So think about if you have a gentleman with a big, hairy beard. So that individual with the big hairy beard, that face, you can think of it as rough endoplasmic reticulum. It is kind of bumpy, kind of scratchy, but when that individual, right, that gentleman shaves, and now his face is baby smooth, right? It's almost like baby skin. That is considered the smooth endoplasmic reticulum. There's no ribosomes that are attached. It's smooth in its surface. Okay, now the function or the job of the smooth endoplasmic reticulum is that it makes lipids. And remember, lipids are fats. So the function or the job of the smooth endoplasmic reticulum is that it makes lipids. Let's now talk about the Golgi apparatus. The Golgi apparatus serves as the shipping center of the cell. In class, we talked about it being kind of like the post office. So the job or the function that the Golgi apparatus or Golgi body may have is to sort proteins made in the rough endoplasmic reticulum and place them into vesicles or containers. Just like we go to the post office and we put things in envelopes and packages and boxes, that is the job of the Golgi apparatus. It sorts these proteins that are made in the rough endoplasmic reticulum and places them into containers that are called vesicles. Let's talk about lysosomes. Lysosomes serve as the recycling center of the cell. And the job of the lysosome is that they contain enzymes that break down molecules. Remember from previous lessons, enzymes help chemical reactions take place. It can either speed the chemical reaction up or it can slow the chemical reaction down. But remember that lysosomes, they serve as the recycling center of the cell, and their function or their job is to have enzymes inside that break down molecules. So thank you, lysosomes, for breaking down those molecules. Let's go to probably one of my favorite organelles is the mitochondria. The mitochondria serve as the power plants of the cell. And so they create energy through cellular respiration. And mitochondria are very special. They are unique compared to other organelles because they have the ability to self-replicate. So they can make copies of themselves and they have their own DNA, deoxyribonucleic acid. So mitochondria is known as the powerhouse of the cell or the power plant of the cell. Their job is to create energy through cellular respiration. Mitochondria are special because they are able to self-replicate, so they can make copies of themselves and they have their own DNA. Let's now talk about vacuoles. Vacuoles serve as storage containers of the cell, which is pretty cool. Their function or their job is to store food, water, minerals for the cell. Okay, so vacuoles are storage containers of the cell. Their job or function is to store food, water, and minerals for the cell. The nucleus, the brain, the control center. The job of the nucleus is to tell the cell how it should be structured and what it should do. Again, the nucleus is where that genetic information is found in the form of DNA, deoxyribonucleic acid, and it is the brain of the cell. So let's talk about the structure of the nucleus. The nucleus has several things that we're going to take a look at. The nuclear envelope, nucleoplasm, DNA, and a nucleolus. So the nuclear envelope is a membrane that separates the nucleus from the rest of the cell. Just like the cell has this semi-permeable phospholipid bilayer, we have a nuclear envelope that surrounds the nucleus. The nuclear envelope is a membrane that separates the nucleus from the rest of the cell. Nucleoplasm is a watery gel-like substance that lives inside the nucleus. Okay, so that's your neoplasm, a watery gel-like substance that is inside the nucleus. DNA, this is found within the nucleoplasm and is the master control center of the cell. Remember, we get our DNA from our biological parents. We get 23 chromosomes from our dad, 23 chromosomes from our mom. Next, let's look at the nucleolus. The nucleolus is a structure where ribosomes are made. So this is fascinating. We talked about ribosomes before. We know that they're important for making proteins, but the nucleolus is where these ribosomes come to life. This pictorial that you see is kind of a labeled diagram of the different organelles that we've talked about. Notice we have our vacuoles, our ribosomes, our mitochondria, our smooth endoplasmic reticulum, the lysosomes, the plasma membrane, the Golgi apparatus, the cytoplasm, the nucleolus, the nucleus, and the rough endoplasmic reticulum. If you are taking notes, I would star this and I would really make sure that I understand these different organelles because this is definitely test question material. So please take the time to review this, and it's really especially great if you're a visual learner. This is going to reinforce the notes that you took about the structure of the cell. I challenge each of you to maintain a questioning attitude, stay curious, and remember, you can do whatever you set your mind to. Thank you for your attention.